The immune system is a complex network of cells and organelles that protect the body against diseases. Cancer cells, through changes in their structure and function, can evade the immune system and survive. Understanding the evasion mechanisms of cancer cells is crucial for developing effective cancer treatments. These treatments are designed to block the evasion mechanisms of cancer cells and trigger immune system activation. Cancer cell development. The development of a cancer cell is a complex, multi-stage process. Normal cells undergo controlled cell division, but cancer cells lose this control capability, and cell division becomes uncontrolled. The cancer development process can be caused by many factors, including 1. Genetic mutations. 2. Exposure to carcinogenic substances. 3. Viruses. Cancer cell transformation results from DNA mutations accumulating in a cell. Cancer cells lose the ability to self-regulate and divide without limit. Normal cells exhibit contact inhibition, where cell division stops when cells become crowded. Cancer cells lose contact inhibition, allowing them to continue dividing and forming disorganized masses. Neoantigens and cancer cell expression. What are neoantigens? Neoantigens are mutated proteins uniquely expressed on the surface of cancer cells, arising from genetic mutations within these cells. Formation process. Genetic changes during cancer development result in the production of abnormal proteins, creating distinct markers on cancer cell surfaces. Immune system recognition. The immune system can recognize neoantigens as foreign and mount a response against cancer cells expressing them. Therapeutic importance. Understanding neoantigen expression is key to developing immunotherapies that harness the immune system to fight cancer, despite cancer cells evolving mechanisms to evade these defenses. Methods for identifying neoantigens. Identifying neoantigens enables the production of vaccines to stimulate immune responses against cancer cells. These vaccines target specific neoantigens present in cancer cells. The process begins by taking samples of cancer cells and blood cells from the patient. Synthetic peptides mimicking parts of the neoantigens are produced and injected as a vaccine to the body, thus activating the immune system response and stimulating lymphocytes, B cells to produce antibodies, and memory cells. What is angiogenesis? Angiogenesis is the formation of new blood vessels hijacked by cancer cells to supply oxygen and nutrients for rapid growth. Chemical signals released by cancer cells to stimulate blood vessel growth develop as a result of angiogenesis, providing pathways for cancer cell spread. Antibodies blocking cancer growth factors. Antibodies like Herceptin and Evasion block growth factors produced by cancer cells. These antibodies prevent growth factors from binding to receptors on cancer cell surfaces. This blockage stops growth signals, halting cancer cell division. Herceptin and HER2 receptors. Overexpression of HER2 leads to rapid cell growth. Herceptin binds to HER2 receptors, preventing growth signals and activating the immune system to target cancer cells. Targeted therapy reduces cancer recurrence and improves survival rates. Immune checkpoint inhibitors, CTLA-4 and PD-1. CTLA-4. Inhibition, specific antibodies, enhances T-cell activity by blocking CTLA-4, which regulates T-cell response. PD-1 inhibition. Blocking PD-1 by targeted antibodies removes suppression on immune responses, enabling T-cells to attack cancer more effectively. Vaccination with specific cancer cell. Antigens. Neoantigens are proteins unique to cancer cells not present in healthy cells. By immunizing the patient with neoantigens, we are activating the immune system to recognize and attack cancer cells. 3. Development process. Identify tumor-specific neoantigens. Produce personalized vaccines targeting these antigens. Vaccinate patients to activate T-cells against cancer. T-cell activity and suppression. 1. T-cells fight cancer. They identify and eliminate cancer cells. Cancer cells suppress T-cell activity, evading immune responses and promoting growth. The suppression by cancer cells hinders treatment effectiveness. CAR T-cell therapy. Cells are modified in labs to recognize cancer antigens. Modified cells are equipped with receptors to target and destroy cancer cells. The modified T-cells are returned to the patient body by IVF. This method treats cancers like acute lymphocytic leukemia and non-Hodgkin's lymphoma. Immunotherapy by immunotoxins antibodies linked to a cytotoxic agent on their Fc leg are targeting cancer cells. The immunotoxins bind to cancer cells, releasing toxins internally to destroy them. But this method requires endocytosis, which some cells resist. Immunotherapy by immunoenzymes are antibodies linked to enzymes that convert non-toxic substrates into toxic products outside the cell. They are effective without cellular uptake and minimizes off-target effects. Steps in receiving immunoenzyme therapy for treating cancerous tumors. The patient receives an infusion containing immunoenzymes. These molecules distribute throughout the body and locate cancerous tumor cells as well as metastatic cells. The immunoenzymes bind specifically to cancer cells through their antibody component. The patient is then given an infusion of a substrate that is specifically designed to interact with the enzyme linked to the immunoenzyme. This substrate is non-toxic and harmless in its initial state. The substrate spreads throughout the patient's body without causing harm. When the substrate reaches the immunoenzymes bound to the cancer cells, the enzyme attached to the Fc region of the antibody converts the substrate into a toxic compound. The generated toxic compound creates localized toxicity, effectively killing the cancer cells without harming surrounding healthy tissues. The battle against cancer involves understanding the intricate mechanisms by which cancer cells evade the immune system and leveraging this knowledge to develop innovative treatments. Cancer cells utilize strategies such as immune checkpoint inhibition, neoantigen alteration, and angiogenesis to thrive and spread. Cutting-edge therapies, including neoantigen-targeted vaccines, monoclonal antibodies like Herceptin, immune checkpoint inhibitors, and CAR T-cell therapies, are revolutionizing cancer treatment by enhancing the immune system's ability to recognize and attack cancer cells effectively. Despite these advancements, challenges remain, such as overcoming cancer cell resistance mechanisms and ensuring therapies are highly targeted to minimize side effects. The emergence of personalized medicine, particularly immunotherapies tailored to individual patients, represents a promising avenue for improving outcomes by integrating genetic insights, biotechnological innovations, and a deeper understanding of the immune system. The future of cancer treatment is increasingly precise and effective, offering hope to millions of patients worldwide.